Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. And welcome and thank you, SynBYSS people, for joining us, the 143rd seminar, and I call it a Harvard Medical School Day. Since I officially joined J. Craig Venter Institute on May 1st this year, I haven't stayed here in La Jolla more than 1 week until these past 2 weeks. Finally, since October 12, I haven't traveled, but I have attended some conferences and hosted some guest speakers at my institute, including Matt Pavlovich yesterday, the Trends in Biotechnology chief editor. It has been great to settle down finally after 6 months of traveling over summer, spending more in-person time with my lab members and institute colleagues. I also look forward to meeting with many colleagues and friends, including today's Rising Star speaker, Dr. Joe, next week here in San Diego, who will attend AICHE meeting, and also my institute's mini symposium that I organize. Okay, it is my tremendous honor to introduce very briefly, I will try, our pioneer speaker, Professor Seth Rakoff-Nahoum. Seth is a professor at Boston Children's Hospital and also Harvard Medical School. I believe I met him last year for the first time in Korea, where both of us attended a microbiology conference in Korea. Since then, we met, I believe, 3 times at 3 different conferences. I really liked his presentation. And when I invite him to be a pioneer speaker, an invited speaker at the microbiome conference that I organized, he kindly accepted my invitation. And that's why, we met, I think 3 times since that. Also, I also remember very bright, his kid attended the conference, a very young kid I'm talking about, I think the elementary school kid, his child, even asked very nice, insightful question to the speaker during I think the Golden Conference at the time. So that was very impressive. So I kind of very delighted to see all the young next generation future leaders, including his own child at the time. And then it's my great pleasure to have him again today. So Seth, thank you so much for your scientific contribution to the community and also to your own child and children, and also for bringing your children to the conference where one of them, as I mentioned, asked brilliant question to the speaker at the time. That basically enlightened us during the conference, I believe. So Seth, the virtual podium is all yours now, and thank you again.
Oh, Taesa, thank you so much for the invitations and for the introduction, but mostly for your kindness since we met right away. I'm honored to be here. I wouldn't call myself a pioneer, but if you want to, that's fine. I wasn't sure if you wanted me to should share some slides or just
It's up to you. It's up to you.
Just talk. Okay.
No, if you want to share the slide, you allowed to share.
Yeah, sure. I'll share some slides.
Sure.
It helps to I prepared some just to, be on the just in case.
Good.
Yeah. Um, so, and also I have like 5 slides, and I always have trouble coming up with a title for what my lab does, but I came up with a term which is mechanistic evolutionary ecology of host-associated microbiomes, and maybe that will come through um, in what I did, what our lab does. And so, what I'd like to do is both, if the goal is to inspire people, let me at least tell you a little bit about my personal history, which basically leads my group towards the questions that we're interested in addressing and how we address them. And that's what I'll talk to you about today. So, so one thing is, and Tessa, I was nervous about talking. One, because I'm always nervous on Zoom. And two is because I think, anytime that you're talking to a group of people where you don't necessarily find yourself as a member of that group. So this is synthetic biology, microbial engineering. I would not call myself any of those, although apparently we do those things in the lab. But within that is, let me just say that my PhD is in immunology, and also I'm a physician scientist. So and I'm going to integrate that a little bit, but I'll say up front is that my PhD began with the discovery of coming right after the discovery of toll-like receptors, which were how hosts recognize microbes and structural components of microbes. And some of these structural components are lipopolysaccharide and fragments of cell wall and peptidoglycan and flagellin. And there are genome-encoded receptors in eukaryotes that bind these and basically activate host defense responses, and in particular are the go signals to adaptive immunity in vertebrates. And within that is my PhD advisor, Ruslan Mezutov at Yale, discovered these with Charlie Janeway. And that's where I really entered the microbiome, which was pretty much a black box of we knew things were there. How did we know things were there? Well, I won't go through the history of that, but we knew that the microbes that live on us weren't necessarily pathogenic, and that they had an influence. And most of that came from studying germ-free mice versus mice that had a microbiome. But mechanistically, how the microbiome was impacting their host was really not known, let alone what microbes were there. And so my introduction to the microbiome came from the lens of innate immunity, which is it's basically a bunch of structures that are recognized by the innate immune system. So that was what my PhD was in. And my PhD focused on really this. So if you can imagine that all the things that I have, listed up here, LPS, CPG, flagellin, those are very common to microbes, regardless of whether they're pathogens or not pathogens. And the recognition of these by the innate immune system, those drive inflammation and immune responses. So my PhD was about, well, okay, now that we know how this is working, how are we not on fire, literally, with inflammatory and immune responses to the microbes that colonize us? Great. So that was my PhD. And because I was an MD PhD student, which of the people that are here today, I at least know that there's one MD PhD in the panel because they're in my lab by Eric. Within that is, and that's funny because I didn't even know he would be here or that he knew about SynBYSS. But within that is what happens is you get your PhD and you're fully in science, and then you have to disappear and say goodbye to science, and now I'm going to go get clinically trained. And what happened while I was getting clinically trained for like 5 to 6 years before when I had my vacation from science was next generation sequencing. And from that, we learned the names of all the microbes that are colonizing or that are present on the earth, let alone on us, in soil, like Jae Sung is going to tell us about. And then we learned all the genes through metagenomics, and then multiple multiomics has told us the proteins and the small molecules and the sugars. And this has really transformed. There's so much biological material there, basically. There's a lot of potential for the effects on the host, let alone for understanding this ecosystem. And basically, because sequencing is cheap and multiomics is decently cheap, anybody that has any money can do a study to associate a disease with the microbiome. And basically from normative processes, physiological processes, and pathology and disease, there hasn't been a disease where there's not a difference in the microbiome. And that partly, and I won't go off on this, is because it winds up becoming epidemiology, and epidemiology always has, differences. And now really we need to understand cause and effect. And so back to a little bit of my career path, when I came back, so all these things are percolating and you say to yourself, well, did I screw up? Yeah. Well, okay, like you're away from science and you say, well, what do you want to know? And what do you want to do if you're going to stay in this field? Because when I was choosing my postdoc, I was like, oh, okay, I'm interested in evolution. Let me go study bird calls. Or I'm interested in pediatric health, but we know that environment influences us across generations. Let me go understand epigenetics. But then I was like, you know what? And this is, I'll leave this as a pearl, not to talk too much of people that are in their careers, which is like, how do you decide the next steps in your life? And I think that, in studying evolution, what we learn is that evolution adapts at the edges. And so I basically said, don't take a big jump, but basically move on the edges. And my decision was to move on the edge and to stay within the gut and the human microbiome and the mammalian microbiome, but to move basically laterally for different ways to answer questions here. And I'll skip through this really because, the questions that we have in the field, and this is where synthetic biology and microbiome engineering collide with other aspects of studying microbiomes or microbes, which is to understand causality and really to understand mechanism. And in understanding mechanism, we then really overlap with synthetic biology and engineering. So my group overarching is really to understand the connections between the microbiome and health and disease, understand which microbes, and then really mechanisms. And then if we can understand the ecology of the human microbiome, we can change it. And if we can understand mechanisms, we can change the functions. And so I'm really going to end with these last two slides because I talk a lot and I need to control that. But basically, my lab's basically back to like, okay, now what do you want to do on the edges? You say to yourself, well, the field is moving towards computation and I can barely check my email. So I'm not going to become a computational biologist. And I'm definitely not going to become a YouTuber. But then you say to yourself, well, but there's a lot, we know there's all these bugs in there because NGS has told us this. We need to hold those bugs in our hands. And so we need really good microbiology. And so basically the field needs really good, really environmental microbiology, which is to isolate these microbes and study them. And when I say study them, I mean study them as much as you possibly can, because if they're important in health and disease, we really need to know the basics, the physiology of these microbes. The next problem, and this I think is the most apropos for this audience, is that we need, how do you study them? And so all these microbes, and I think, I'm imagining that Jae-Sung will tell us about these things because it's very similar in environmental and soil microbiologies. How do you study these microbes, or how do you, harness them when they're genetically intractable or there's too many to study? And so basically we need this to understand physiology, and we need to understand bacterial physiology and really all the microbes that live in our gut, especially the ones we think are important. And so we need to make these organisms, we have to understand their genes and their enzymes. And so that's molecular biology, but we need to do that within the organism itself. And to do that, we often have to make the organism genetically tractable. And so, I think there's a lot of deep biological important questions there. because, if you look at this red arrow, which is a gene which is conserved across all these different microbes, we know as evolutionary biologists and molecular biologists that genes don't function the same within different organisms, even if it's the same gene. And so this is where, I think we need to understand, even at the gene level, how genes function within different organisms. So there's a lot of network biology and synthetic biology, molecular biology, and some, microbiome engineering that's needed in the field. And then I'll really end with, two aspects which I think need to be combined here, which is we need to understand ecology, and use ecological frameworks to understand the forces that shape an ecosystem, and that's environment and microbe and host microbe interactions. And then really, which is, I think, the most fun, especially when you combine it with all these other three approaches, which is understanding evolution. And this is really when we think about the relationship between microbes and their hosts, and we've learned about microbes and their hosts for unicellular organisms, such as chloroplasts and mitochondria, the evolution there, about aphids and endosymbionts, about Vibrio basically symbiosis in like Hawaiian bobtail squid. Maybe we can capture some of those when we're in Hawaii. And then cockroaches and termites. We've learned a lot. Pitcher plants. We learned a lot from basic the field of symbiosis. And so when we think about the parameters of understanding those, we can think about transmission and dispersal and service and function, diversity and niche. And we can think about like what is the parameter space for each of these really fundamental aspects of symbiosis that really need to be understood. Basically, what all kind of come together, all five of these major domains come together in understanding these symbiosis. And then we think about humans, we don't really know. We know we can assign, well, are we an open system? Are we a closed system? How much vertical versus horizontal transmission are there? We know that it's not a one microbe system, it's a many microbe system. When you have a many microbe system in a closed or an open ecosystem, and you think about the functions, it's a mess. But really, what we want to understand, combining ecology, molecular biology, and microbiology, is to understand these eco-evolutionary processes. And that's what my lab does. And we basically take top-down and bottom-up approaches. We take poop to use math and ecological evolutionary theory. Then we get candidates, and then we have the microbes and the genetically tractable organisms to test these things out. The other way is to start with microbes and just ask basic biological questions. And then we learn about gene function and protein function. And then we want to ask their significance within complex ecosystems and the host. And so then we move up. And I'm not going to get necessarily into any of the science, but really this is the generalized questions and approaches of the lab. And I know that I spoke longer than 10 minutes, and sorry for that. And thank you again for the opportunity.
So amazing talk as always. So I love it. So you keep mentioning you are not synthetic biologist or not known to the synthetic biology community, but let me put it this way: you are a pioneer, basically, who dismantle or disrupt the barrier between fields, or among fields. I mean, you basically disrupt or, dismantle this kind of barrier. One clear example I can tell you is hardcore synthetic biologist Brian Huang joined your lab because, you influence those kind of young kids, I mean, to also break those kind of wall between fields. So you are a pioneer, you are known to the community, and then thank you for doing so. And I believe, multiple different, scientists and engineers collaborate, intellectually. That is how we advance science. So thank you so much for you doing so.
Thank you. Thank you.
Yeah. Okay. So now the main speaker of today with a longer introduction, Dr. Jae-Sung Jo is currently a postdoctoral associate in the Chris Voigt lab at MIT. He received his bachelor degree from Georgia Tech and PhD
Oh,
from KAIST under the guidance of Professor Sang-Hyuk Lee in 2021. His graduate work was focused on the development and application of genetic engineering tools for Corynebacterium glutamicum, a very important strain used industrially to produce amino acid and chemicals. His early research efforts were focused on the development of CRISPR-Cas9 genome engineering technique to engineer C-glutamicum. More recently, he developed a reprogrammable synthetic small RNA-based gene knockdown tool that can be used widely, just not only C. glutamicum, but also other gram-positive bacteria to knock down genes and screen gene targets for metabolic engineering. With these molecular tools, he showed that this, bacterium could be engineered efficiently to produce various important chemicals of interest. He also designed and engineered microorganism capable of producing methyl anthranilate, a grape flavoring compound widely used in food. They were the first to report the bio-based fermentative, production of this important flavoring compound to the significant level, and the engineering strategy used can serve as a framework for the production of other volatile aromatic esters in microbes. In addition, he led a team to develop a bio-based process to convert bio-waste to a value-added chemical. His current interests are in the development of synthetic biology tools for undomesticated organisms. When I visited MIT to give an invited talk last year, I also stopped by the Chris Voigt lab without any notice at 10 p.m., I believe. He was there still, 10 p.m. I'm talking about, to work hard. He's a truly rising star with promising future. This year, I visited again, MIT area for the conference. I also treated him with very nice Korean barbecue along with other postdoctoral SynBYSS rising star speakers to cheer up, them basically. So Jason, thanks so much for your time today, and please take it away. Looking forward to it.
Thank you so much, Tesak, and thank you so much, Seth, for the wonderful talk. Today I will talk about my grad school work. So I'm sorry, Seth, I won't be talking about soil or environment today, but I'll talk mostly about this bacteria called Corynebacterium glutamicum, which is, I think, equally very interesting. I think Tesak, Professor Moon has uh, described my academic journey well. I was actually born in Belgium, in a small town called Leuven, and I grew up in Singapore. That's where I spent most of my growing years. I did my undergrad in Georgia Tech. I did my undergrad research in neural engineering, so I was dissecting mice brains a lot, and it took a long time just to get some data. So I was like, okay, maybe I should go into bacteria, which is pretty fast. So I shifted to working in the field of metabolic engineering at KAIST, and I started my postdoc about 2.5 years ago here at MIT with Chris Voigt. So today I'll focus more about the work that I've done during my PhD, which is basically working with this bacterium called Corynebacterium glutamicum. And to talk about this strain, we really have to talk about MSG first. Like the MSG that we eat, we put them in our food, right? And have you ever wondered where we get our MSG? I think a lot of people think that we produce it chemically, But so how did this come about? Where we have to look back into the history of MSG. So in 1908, more than 100 years ago, this professor called Kikune Ikeda from University of Tokyo, he found this white crystal from kombu. It's a type of seaweed. And when he added sodium hydroxide and he tasted it, and he said it gave a beautiful flavor. And he said that he found the essence of flavor. We call it umami now. And his vision at the time was to relieve the malnutrition in Japan because it was a very it was a time where the Japanese people had their lifespan reduced, and so he wanted to find a way to make food more flavorful. And there was this businessman called Saburusuke Suzuki. He shared the same vision to relieve the malnutrition in Japan. And they founded this company called Ajinomoto. This is a very famous company in the food industry because a lot of the food additives and food products are produced by Ajinomoto. So at that time, about 100 years ago, they concentrated HCl to decompose gluten to make MSG. And they did this by producing, they use clay pots to hold concentrated hydrochloric acid. And this was very dangerous. And they had a lot of backlash from the villages when they set up their factories. So they had to move away from the city to do this. And post-war Japan, 1950s, they were trying to, again, relieve the malnutrition of the Japanese. And it was at this time that they were looking for a way to relieve to find a way to really to produce, their vision was to produce food protein from fermentative products. So this was actually a very visionary statement that they made back almost 70 years ago. It is only now that we're actually thinking about producing food proteins using fermentation. And while they were doing this, they actually discovered this bacterium called Corynebacterium glutamicum, 1956. They found that it produced a lot of glutamic acid, and they thought, this is an amino acid, so maybe we could use this, and it's better than nothing. It's not entirely a food protein, but this can pass as a nutrition. So they discovered Corynebacterium. They set up and succeeded the first fermentative production of an amino acid that time. And it was really interesting how they were able to really open up this whole field of amino acid fermentation in 1970s, just because they wanted to solve this problem of relieving the malnutrition at the time. So all of this is actually written in the Handbook of Corynebacterium glutamicum. This is a book that I read when I first started my grad school. It's really interesting because Dr. Ikeda, the one who actually isolated the strain himself, he wrote the first chapter, and he reminisces the past, and he writes about all this story. So I'm more of a book kind of person. So I like to read this kind of science history. And I think it's a great way to really show how the field has evolved and the origins of the field. And so since then, Corynebacterium glutamicum has been used to produce a lot of different amino acids and feed industry. And it's a very important strain in the feed industry. So not only glutamic acid, lysine, threonine, methionine, a lot of the amino acids are being produced by Corynebacterium glutamicum today. So these are some big companies that produce amino acids from Corynebacterium glutamicum. This is a Chinese group, a Japanese group, and a Korean group. And this is an example of a plant that they use to produce them industrially. So the amino acids production, it's a huge market. With MSG, they produce over 1.7 million tons a year. It's a $5.5 billion market. And lysine, also 2.5 million tons with a $4 billion US dollar market. And C-glutamicum produces most of these products. And so Corynebacterium glutamicum, this is a very robust strain. It's a gram-positive strain. It's a facultative anaerobe. It has a very rich history in industrial fermentation. And a lot of the chemicals that are actually all the chemicals that are produced by this strain is labeled GRAS, which is generally recognized as safe. So because the industrial fermentation techniques and processes are already set up, it can be used as a production host for a lot of different chemicals for the bio-based production, for the sustainable production of chemicals. So there were two questions that I wanted to answer during my grad school. The first was, can we develop advanced genetic engineering tools to really push the production limits of this strain so that it can produce a lot better any kind of chemicals in an industrial setting? Because previously people used very traditional methods to engineer the strains. They just use a random mutagenesis. So they don't really look into like the DNA or like the genome sequences to see whether to rationally design the strains. And the second question I wanted to ask was, can we engineer the strain to produce high titers of non-native chemicals that are not necessarily native to the strain itself? For example, glutamic acid and lysine, they are metabolites that the strain produces on their own. But can we engineer the strain to produce non-native chemicals at a very high titer, at a very competitive level as well? So for people who don't know who are new to metabolic engineering is this field where you look into the metabolic pathway of an organism, and you basically engineer the metabolism so that when you introduce a substrate, for example, glucose, you'll be able to produce your product of interest. So how you do it is you either overexpress the pathways that you want to overexpress, or you knock out competing pathways so that the carbon will be directed to the product of interest. So you do this by engineering the chromosome, the genome of the bacteria, by knocking out the genes or overexpressing the genes. And so I'll talk about some tools and the examples that I have done during my grad school as a example for this strain. And so the first thing that when I started my grad school in 2015, we were looking at the genome engineering tools for Corynebacterium glutamicum, and I noticed that a lot of the tools that has been developed were very time consuming, and the editing efficiencies were very low. They had used these very old methods to engineer the C. glutamicum genome. And so there was a need for a very highly efficient and rapid genome engineering tool. And it was around this time where CRISPR was all the rave at the time. A lot of people were applying CRISPR to like all kinds of living organisms at the time. And I also jumped in and wanted to see if I can engineer C. glutamicum easily or faster than the traditional methods using CRISPR. So for people who don't know how CRISPR works, Cas9 basically works in such a way that you have this Cas9 protein. This is a protein, and it binds to a guide RNA where you can program the guide RNA to be a target a specific gene in the genome. So this complex, this RNA and this Cas9 protein complex, they form a complex, and they will scan through the genome until it finds the target gene of interest. And then once it matches that site, it will cut that region of the chromosome. And if you introduce a donor DNA, it would repair it. So this is how it works in a lot of living organisms. And at that time, I was new to the field. And the first thing that we did was we wanted to see whether we can produce the Cas9 protein. Unfortunately, when I introduced the Cas9 protein to C. glutamicum, it didn't work. And I spent several months trying to debug it until somebody told me maybe I should just try using a codon optimized version of it. And when I did that, I was able to express Cas9. So I expressed Cas9, and I was able to have this system working in C. glutamicum. The only problem was that the CRISPR system didn't really work the way it does in other living organisms especially for bacteria, because bacteria doesn't really have a repair system. They do have repair systems, but they're not as efficient as other repair systems for higher organisms. So every time I introduced and targeted the genome, the bacteria simply just died. It didn't. I wasn't able to. I tried to introduce some donor DNA, but it didn't. It didn't work. So what we did instead was we flipped the strategy. So instead of editing it first and then introducing a donor DNA to repair the system, I made it so that I actually edit the DNA, the genome first, and then use Cas9 to screen out those that didn't get edited. So how this works is I introduced a recombinase called RECT. So what this does is it binds to a single-stranded DNA that you want to edit, and it creates this single-stranded DNA RECT complex. So I will first edit the genome first, and then for those that have been edited, it will survive. But those that has not been edited, I'll use CRISPR-Cas9 to cut the genome, and they will die. So only those that have been edited would survive. So this is how we flipped the approach, flipped the traditional method to use it for our bacteria. And I did some protocol optimizations at the time, and I was able to finally get reproducible results by knocking out this gene ARCH-R using this new system. And I completed the protocol. I created this protocol where I was able that we could iteratively engineer multiple genes at the same time by making mutations to the plasmids so that they can be cured easily. And so once all the engineering has been done, you are able to cure the plasmids, both plasmids at the same time, and then you get this mutant strain that has been engineered and that is free of all plasmids. And as an example application, because we applied this to produce GABA. GABA, I think people know it as a metabolite that's in the brain, but it can also be used for nylon synthesis because of its structure. And it's a one-step reaction from glutamate. You just need to introduce this one gene, and you will produce GABA from glutamate. And you know how Corynebacterium glutamate produces a lot of glutamate. So this was it was a good example to show. And there are 3 different target genes that can be engineered. And if you engineer them, you can actually produce a lot of GABA. And so you can see how, with just the introduction of GAPD, you only get a little bit of GABA production. But when you start engineering these genes, you'll see how it really increases the production of GABA. And so we were able to construct these 8 different strains of different combinations of these genes, and we were able to get this Corynebacterium glutamicum strain to produce almost 30 grams per liter of GABA. The point that I wanted to make here was that we were able to construct these 8 strains within 2 weeks. If we had used the traditional method to engineer the strain, it would have taken several months, or it could take a long time. But because we had set up this CRISPR-Cas system, we were able to engineer and knock out these genes really quickly. So that was that for genome engineering. But there's still some problems with genome-based engineering because it's still quite labor-intensive, and you can't really target essential genes, and you can't really target multiple genes at the same time. And every time you want to test out whether one gene is going to be a good target for engineering, you have to actually go through all this procedure to knock it out. And so, for example, if you're a metabolic engineer, you want to produce this compound called valolactam, right? It comes from lysine. It's several steps from lysine. And the question is which genes should I engineer to be able to increase the production of valolactam? And so this thing came up from the question of how can we quickly screen genes that are potential candidates for the overproduction of a chemical. And so we turned to this tool called synthetic sRNAs. Synthetic sRNAs is a technology that we developed about 10 years ago, where this is how a normal transcription translation works. But synthetic sRNA is how you can engineer and reprogram the sRNA scaffold, and it will bind to the mRNA of interest. And so the ribosome will not be able to come and bind to the target of interest. So you get a reduction in translation. And so previously we had used E. coli-based sRNAs, but when we applied this to Corynebacterium glutamicum, we were not able to see a lot of reduction, only about 35% reduction in a gene. We want to make sure that it goes more than 50% to make it viable for use. So we looked at different sRNA systems in gram-positive bacteria. So sRNA systems work by having an sRNA scaffold and a target binding region, and you have this HF-fused chaperone. This is a protein that binds to the sRNA, and it helps bind to the mRNA of interest. So we looked at many different papers. There were a lot of sRNA well studied in gram-negative, but not so much in gram-positive bacteria. So what we did was we looked at a number of sRNAs that we can take from gram-positive bacteria, and we tested them in Corynebacterium glutamicum, and we're able to knock down the genes much better than the original E. coli sRNA system. And so with that, we tried to knock down metabolic genes in Corynebacterium glutamicum, and it seems like it's a viable way to do this, a system to knock down genes in Corynebacterium glutamicum. And so there are two ways to do this. We can use sRNAs to do a rational targeting, and another way is to do a random targeting. I'll talk about how we can use sRNA for rational targeting, and I'll show this with two examples. So again, going back to the example of valorolactam, valorolactam is a compound that's used as a monomer for polyamides. And so you have this pathway, right? And so what we did was we constructed the gene targets So these are some potential gene targets that we know that could enhance the production of valorolactam by directing the carbon flux to lysine and then to valorolactam. And so we constructed all the genes. If you take the traditional way, you would have to knock down each gene to see whether they actually affect the production of this. But if you use sRNAs, you don't have to do that. You just construct them in plasmids, and then you introduce them to the strain. And then with that, you can quickly see that when you knock these three genes down, they actually increase the valorolactam production, right? So this is one way to really quickly screen instead of going into the genome and knocking out all the genes one by one to see which one has an effect. So you can use sRNA in a plasmid form like this, and we confirm that we confirm the production of that we confirm the knockdown of it with this. And then another example is methylenthranilate. This is a grape flavoring compound. I'll talk more about this. And again, we did the same thing where we looked at potential targets, and then we introduced them with an sRNA, and then we did the sRNA-based repressions, and we found that there are some of them that actually increases the production of methylenthranilate. And we took a step further by actual engineering of the genome, not only the plasmid reduction, but we also engineered the genome so that it will be reduced. And we found that when we knocked down these genes, we were able to get better production of our compound. So that's how you can use sRNA for rational targeting. But you can also use it as random targeting that targets like the entire genome. And I'll talk about this with an indigoidin. This is a viable alternative colorants that is used in that gives a blue color, like indigo. And so how you can do is you get a pooled oligonucleotide library that targets all the genes in the genome, and you clone it into the siRNA plasmid vector, and then you get a pooled library, and you transform it into the target producer strain, and you can screen them. And so that's how we did it for this chemical, where we pulled the library and we introduced that, and then we were able to see that 13 colonies showed over 5-fold increase in indigoidin production. But then we wanted to make sure that this was not due to colony variation, but that it was actually indeed by the downregulation of those genes. So we re-engineered and recloned the plasmids and reintroduced them, and we showed that it was indeed that 5 of them were indeed helpful for the indigoidin production. So the point that we wanted to make here is that when we use this siRNA pool library system, we were able to find targets that we would otherwise miss, because these are just proteins, like membrane proteins, that we would never ever have imagined or thought that would affect the production of indigoidin. But because we did this and we screened the entire genome, we were able to find targets that are non-obvious for the production of indigoidin. So does this system work in other bacteria? We tested this across various bacteria, and we were able to show that this works in other bacteria as well. So this is a universal knockdown system that we can use to knock down genes on the mRNA level. And it also works to help alterate phenotypes in different bacteria. And so that was the tools that I developed. And I will really quickly talk about the chemicals that you can produce with Corynebacterium glutamicum using these tools. The first was the methylantranilate that I briefly talked about. This is a chemical that gives off a grape flavoring. So when you grow this strain, it actually smells like grape. It's fascinating how it has such a potent smell. So this is used in like our grape juice, and they have to be so all the grape juice that we have to be labeled as artificial flavoring, because the current synthesis for methylantranilate is from fossil fuels, and we want to make sure. So what we wanted to do was we wanted to produce this in using biological methods, and so there are 2 pathways to produce this. And this is known usually in plants. There are two pathways, but we decided to go with the simpler pathway because this pathway requires methanol, and I don't think a lot of bacteria would do well in methanol. And this other pathway is a SAM-dependent pathway, and SAM is S-adenosyl methionine that is quite common in a lot of organisms. So we decided to go by this route. And we engineered first the E. coli to produce this chemical, and we used similar techniques to engineer C-glutamicum as well. And so very briefly in E. coli, we first introduced the gene, and then we set up this 2-phase extraction system because the MANT, this is a chemical that is not soluble in water. So we had to introduce some sort of an oil-based oil that can help extract the MANT as it's being produced. And so we did a lot of optimization to increase the production of MANT, and we were able to get about 4 grams per liter of MANT in E. coli. And so here, this is where we had the aqueous phase and the organic phase. The organic phase is we use tributyrin to extract in real time the MANT from the bacteria. And so this really is advantageous because you don't have to do downstream extraction because it's already extracted straight to tributyrin. So this is one of the ways that we did to extract the compound of interest. And we got the gene from corn because apparently corn also produces some level of grape smell as well. And so I'll just really quickly go over this. And we were able to produce a high titer for that. And we did something similar in siglutamicum, almost everything. But the pathway engineering is slightly different for siglutamicum. But I'll just talk about the last part. So from ANT to MANT, for E. coli, we had to make sure to introduce engineered so that we have a lot of the SAM, the cofactor for this. And so we engineered E. coli to produce, we even supplemented methionine so that it will go down this way. But when we did that for Corynebacterium glutamicum, it didn't really work well. So what we did instead was we did a recycling, we enhanced the recycling pathway, and that actually really increased the production of methylenterinylate. So for different bacteria, we need to take different approaches to engineer them, because I think each bacteria has their own regulatory, different native regulatory networks to to produce the product of interest. And so we optimized the fermentation, and we were able to produce a lot of methylenterinylate. And this is a very competitive a number for a chemical like this, because it's very difficult to produce fine chemicals like this. And another more recent chemicals that I wanted to share, and this is a work that has been accepted and it will be published very soon, is looking at pseudoaromatic dicarboxylic acids. So you know how, like, when we look at PET or PBT, we have all this moiety, this aromatic dicarboxylic acid moiety. So basically it's an aromatic ring with a dicarboxylic acid on both sides, and you combine it with ethylene or something else, butylene, you get these polymers that you use to make PET or PBT or a lot of other plastics. And so the question arises from, can we produce bio-based alternatives to these aromatic dicarboxylic acids? And so currently there are these petroleum-based monomers, right? But there are also chemicals that look very structurally similar that occurs in nature, right? For example, terephthalic acid looks very similar to 2,5-pyridine dicarboxylic acid. It's the same aromatic, but then this is not necessarily an aromatic, so we call them the pseudo-aromatic dicarboxylic acids. And some of these monomers, they occur naturally when some bacteria degrade lignin-derived aromatic compounds. So and especially PDC has been successfully polymerized with various diols and hydroxy acids to really create a diverse polymers that have never been seen before. So if we're able to utilize this to make polymers, I think we can really open up a whole new class of biomaterials that we have never seen before, with very interesting and novel physical properties. And there's remarkable inactivity of using them in polyester synthesis. And I think it is only now that people are starting to look at these chemicals as an alternative to bio-based terephthalic acid. And if we use this, actually, we can also create biodegradable plastics, because they have shown that PDC, when they combine it with other, when they used it to make polymers, they were able to degrade it within a few months. And so the challenge is that current methods to create these pseudoaromatic compounds is very difficult, because you need a lot of it's very low yield, and there's not much the precursors are it's not available, and you need very harsh reaction conditions at high temperatures, and you need to use a lot of strong bases. And so we wanted to take this using a biological system where you just need a single bacteria and you ferment it at 30 degrees. And all you have to do is you introduce a single substrate, glucose, and you're able to produce all these different interesting compounds. So this is the overview of how we engineered Corynebacterium glutamicum to produce all five different pseudoaromatic compounds. But very briefly, we constructed a base strain that's able to produce a lot of PCA, because a lot of these are derived from the shikimate pathway with PCA as the intermediate. And so we did some metabolic engineering to produce PDC first by plasmid-based expression of the genes that is used to produce PDC, and we also did genetic manipulations by knocking out in different combinations of all the genes that may be required to produce this. And we were able to produce about 10.89 grams of PDC in flask. And so we did a lot of fermentation process optimization. We did like delayed induction, and this is all done in a 5-liter reactor. And we did reactor engineering, and further transcriptome analysis-based engineering. We were able to produce this at 76 grams per liter, which is a very high titer for such a novel compound. And so we're very excited with this data. And so using this strain, we use this strain to produce the other pseudoaromatic compounds, because by just changing the last gene, we can able to produce different compounds. So instead of using the genes to produce PDC, we introduced this PMDAB and LIGAB to produce this 2,4-PDCA. And for 2,5-PDCA, instead of using these genes, you just have to introduce this gene to produce 2,5-PDCA. And because they're all cleaving the aromatic ring and then putting it back together using ammonium chloride, we thought, 2,3-PDCA can also be done in the same manner. So this is actually a de novo pathway that we looked at the pathways for 2,4 and 2,5-PDCA, and we thought, they kind of look similar. So maybe if we add ammonium chloride, we'll be able to close this ring and create 2,3-PDCA. And so we actually proposed, and actually we demonstrated this new pathway for 2,3-PDCA. And so we were able to produce quite a substantial amount of all these pseudoaromatic compounds. And the last thing that we wanted to do was the 2,6-PDCA, just to complete the portfolio. And so we engineered another strain that is going from aspartate, and we introduced and knocked down many genes, and we were able to produce 15 grams of 2,6-PDCA. And so this was my little contribution to Corynebacterium glutamicum, this really important and exciting strain that is used in industrial fermentation. I developed some tools for advanced metabolic engineering using CRISPR and synthetic sRNAs. And I engineered the strain to produce a number of chemicals, and to show that this strain can be used to as a production platform, like a small little factory to produce a lot of very interesting chemical compounds that can be scaled for the sustainable production in the industry. And so I would like to thank my graduate advisor, Sang-Yep Lee, and all the people who have helped me during this time of my grad school. And thank you.
So amazing. Thank you so much. It's amazing, and looking forward to reading your very fresh new paper, that is out soon, I guess. So let me ask some question.
Yes.
So actually two question. One is a little bit technical question. So
Yes.
you show one slide earlier about GABA production,
Yes.
and you knock out, I guess, GABA, I think, importer.
Yes.
And I just wonder whether that GABA importer only working one directional, like, importing only, importing, and then why you knock out if that is only one directional kind of transporter?
Um,
Because
I
I mean,
think
I mean, if you knock out that one, if that is only importing,
yes,
GABA somehow accumulated, but of course GABA probably going out as well, by whatever passive diffusion, whatever.
yeah,
And then, you knock out also the, consumption pathway, and
yes,
then accumulate means, chemical, equilibrium prevent production more, right?
right, right,
Chemical, perspective. So I just
yes.
wonder why, I mean, that is the case.
Okay, so I think that's a great question. I think so this was quite some time ago, but I think at that time they actually discovered that this GAP gene was responsible for the importing of GABA. And not necessarily just GABA for I think similar compounds, but if you knock it out, so I think it works as an importer, not necessarily as an exporter as well.
Hmm.
So I think that's why when we knocked this out, we actually saw an increase in the production of it. It's not
Oh,
a
okay, okay.
huge,
You kind of
yeah,
quickly go, so I didn't, clearly see the graph.
yeah.
So that, knockout basically improved the titer
Yes,
somehow.
yes, yes,
Oh, I see.
yes.
Okay, that makes. But do you know what is the real function of that, transporter? This kind of only one directional?
I'm not very sure.
Okay,
Yeah,
okay, I see. And
yeah,
then, for the last part of the your talk that will
yeah,
be published soon,
yes,
uh, so you kind of make very interesting compound,
yes,
and there are one period of time in metabolic engineering, people try to make, kind of very interesting new compound. And then, but at the same time, I kind of wonder, those compound could be useful in the future. But at the same time, we need to create some new market, utilizing this interesting kind of molecule, later on,
yeah.
rather than just kind of showing, oh, we produce some interesting, compound, and then publishing some paper. So, what would be your perspective, regarding future metabolic engineering field? People talking about we can make any compound using metabolic engineering. That's kind of same promise, organic chemist, already, mentioned, many, decades ago, we can make any compound in the world. And then that's how, currently, pharmaceutical company doing, organic chemist, synthesis to make bunch of different small, whatever molecule or big molecule, small molecule, whatever, and then test high throughput way to, identify some drug candidate.
Mhm.
But if you can do that same way, I mean, in the future, that's amazing. But at the same time, we more focus on bulk, production, large scale production. At the end of the day, but we need to create some utility of this interesting molecule in the future. I just wanted to hear from you.
Right. I think that's a very important question, I think. And I think just taking the example of PDC, so
Mm-hmm.
this is a chemical compound that is degraded from, shingobium. And the problem is they wanted to study this as a material, but the problem was that they were not able to produce this enough to study it as a chemical. So I think there are a lot of very interesting chemicals that have very, put huge potential as new material. Is I think the problem is actually having enough of it to study the physical properties.
Mm-hmm.
And in order to do that, we need to scale it up first and
Sure.
produce enough of it so that we can extract it and purify it and use it as a chemical for polymer studies or things like that. Because when you're actually making polymers, you need a lot of it to make the polymer reaction happen.
I see. I see.
Yeah.
So you kind of believe in the future, considering economics of those fermentation, uh, now in the future, I don't know when, but let's say 10 years from now, Sigma Aldrich completely replace all the chemical they selling, but producing everything from fermentation. You think that happening?
I'm not so sure if it's going to be 10 years, but I think that is the future. I think we have to use bio-based methods to make them because it's really, I think it has so much more advantage than using chemical
Sure.
synthesis. Yeah.
Sure.
Yeah.
Absolutely. So beautiful work. I'm looking forward to again to
Yeah.
reading your
Yeah.
paper that published. Uh,
Thank you.
so great. So let me check whether we have question from audience. Okay. Okay. So unfortunately, the SES need to go to another meeting,
Yes.
11 Central Time or noon,
Yeah.
Boston time. So I don't see any question, now. So also we are already past the 11:12,
Right.
the Central Time. So let me close and then we chat afterward.
Okay.
Okay. So thank you all for joining us and staying with us today. We'll meet again on October 31st. Oh, Halloween. Thursday, the same time, the same Zoom link. We'll have Dr. Tim Lu, used to be at MIT, but now at Senti Bioscience, and Dr. Emil Damgaard Jensen at Technical University of Denmark, or DTU. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thanks. I will stop recording. Just give me 1 second.